A Potato Made with Gene Editing by Antonio Regalado from Technology Review Magazine, June 2015. Dan Voitas is a plant geneticist at the University of Minnesota, but 2 days a week he stops studying the fundamentals of DNA engineering and heads to a nearby company called Selectus Plant Sciences, where he applies them. His newest creation, described in a plant journal this month, is a Ranger russet potato that doesn't accumulate sweet sugars at typical cold storage temperatures. That will let it last longer, and when it's fried, it won't produce as much acrylamide, a suspected carcinogen. What's different about the potato is that it was bred with the help of gene editing, a new kind of technique for altering DNA that plant scientists say is going to be revolutionary for its simplicity and power. The technology could also be a way to engineer plants that avoid the stigma and the regulations normally associated with genetically modified organisms, GMOs. In the case of the Ranger russet, Voitas's gene editing technique, known as TALENs, left behind no trace other than a few deleted letters of DNA. The edit disabled a single gene that turns sucrose into glucose and fructose. Without it, Voitas thinks, the potatoes can be stored far longer without loss of quality. The potato is a prototype of what plant scientists say is a rapidly arriving new generation of genetically modified plants. With gene editing, small companies think they can very quickly develop new crops for a fraction of the typical cost, even in species so far mostly untouched by biotechnology, like avocados, sorghum, and decorative flowers. Most genetically modified crops that have been grown commercially so far incorporate genes from bacteria to make them produce insecticides or resist weed killers. Public opposition and regulatory requirements make these transgenic plants expensive to develop. That is why nearly all biotech plants are lucrative, big acreage crops like soy, corn, and cotton, and are sold by just a few large companies like Monsanto and DuPont. In August, the U.S. Department of Agriculture told Selectus that, unlike transgenic plants, its potato wouldn't be regulated. That means instead of being grown in fenced-in test plots and generating folder upon folder of safety data, the Ranger russet may go quickly to the market. 2 years ago, the agency reached a similar conclusion when it considered a DNA-edited corn plant developed by Dow AgroSciences, although it isn't being sold yet. Scientists say products like the potato are just the start for gene editing techniques in plants. The same techniques are going to allow far more sophisticated engineering, including manipulation of photosynthesis, to make plants grow faster and yield more food. It's an enormous opportunity, an unfathomable opportunity, says Martin Spaulding, a plant researcher at Iowa State University. For now, the techniques are being used to modify plants in more modest ways. The first wave of this technology is just removing a few base pairs, says Yinan Yang, a professor of plant pathology at Penn State University, referring to the combinations of DNA letters, A, G, C, and T, that make up a genome. By knocking out just the right gene, as researchers did with the potato, it's possible to give a plant a few valuable properties. The next step, Yang says, will be to change the DNA letters of plant genes, swapping one plant's version of a gene for that of another known to offer, say, resistance to disease. Yang says there is a blight-resistant form of rice that differs from commercial species by only a few DNA letters. I could just change that over to resistance, he says. It's like gene therapy in humans. He says he's negotiating a contract to produce the gene-edited rice now. As for Voitas, this isn't the first time he has set out to gene-edit plants. A decade ago, he started a company called Phytodyne, based on an earlier technology called zinc finger nucleases, but it folded after Dow AgroSciences paid more than $50 million for exclusive rights to use that type of gene editing in plants. Voitas teamed up with the French biotechnology company Selectus in 2010 after it offered to install him as science chief of a new plant engineering division. But initial efforts ran into difficulty when another gene-editing system, meganucleases, proved challenging to work with and also got tied up by patent disputes. Eventually, Voitas returned to the lab and co-invented a new way to edit genes using specially engineered proteins called TALENs. That technology was used to make Selectus's potato, as well as a soybean with improved oil. Since then, Voitas and Selectus have also worked with a newer technique called CRISPR.